Hi, and thanks for watching. Today I'm presenting work mostly done by an amazing team of students that I had the chance to work with during my postdoc at Michigan State University. Parasites are everywhere, and we know they can have very strong impact on their host. Today I will tell you more about Schistocephalus solidus, a tapeworm with a complex life cycle. Their eggs are found in bird species. If they land in the water, they'll be eaten by copepods, which in turn will be eaten by fish, and the eggs will develop in the fish's body cavity. The fish will be eaten by birds, and the parasites can reproduce in the bird's bloodstream. The worms can develop to be as big as the fish body cavity or more, and there can be lots of them in there. So you can imagine that they can have very strong consequences on the fish physiology, but also on their behavior. So it's been shown multiple times that the parasites seem to be able to modify the fish's behavior so that they are more likely to be eaten by birds, which is the parasite's final host. So you'll see the fish swimming to the surface of the water, and they will seem to be much less afraid of birds as well, so they're more likely to be eaten. We're working with fish from Iceland, and so we have been collecting sticklebacks in 15 different populations all across the island, in the highlands as well as lowland lakes, and in marine populations as well. Lucas Prashun has been screening fish from 15 different populations for all types of parasites. He's looked at 578 fish and found 70% of them to be parasitized with at least one type of parasites, 12% with a solidus, and they generally had 1 to 2 worms in their body cavity, but up to 20. Brooke Harper has been looking at how this might affect reproduction by measuring ovary size in all those fish. And here for a subset of population, I'm showing you relative ovary volumes, so controlled by their body size. And you can see in green, these are parasitized fish. She found that they have smaller ovary size than non-parasitized fish in purple. And this was sometimes a very big difference, like here in Christie, and sometimes no difference for some of these populations. Looking at their behavior, I've been analyzing results of behavioral trials where we simulated predation, in this case with olfactory cues. So there's a smell of danger in the water. And in a lot of the cases, fish will react by freezing. So they stop on the spot and they look around because they don't know where the danger is coming from. But what I found is that parasitized fish are much less likely to freeze than non-parasitized fish here. So this is another confirmation that the parasites are affecting their anti-predatory behavior. Elizabeth Phillips, who is now a PhD student at Wageningen University in the Netherlands, has been interested in if parasites might affect the fish senses and vision in particular, by showing the fish pictures of food items, in our case bloodworms, and she had pictures of increasing sizes here and was noting when the fish would first react to the moving picture of a bloodworm. And so these are 2 different populations, and you can see here for this Christie population, parasitized fish in green are much less likely to detect the food at all, or they will only do so when the worms are really big. While for this other population, Galta, parasitized fish seem to be able to detect even the smallest size worms. So it seems like parasites might be able to affect vision in one of these populations. McKain Williams has been looking at the brain size of these fish. And here I'm showing you relative brain volume, controlled by body size again. And what you can see, these are the 2 same population. And for this Christie population, where we just saw that fish had trouble seeing the worms, it seems that parasitized fish have much smaller brains. Now, there are a lot of differences between these 2 populations. And I would be more than happy to talk to you more about some of the ideas we have as to what might influence these patterns between populations. In summary, I've been showing you that parasites affect reproduction, behavior, they might affect senses and even brain development in sticklebacks. And so future step will include analyzing brain regions in more details, candidate physiological pathways. This is in collaboration with Hans Hoffmann's team in Texas. But also I would really like to look more into those environmental conditions that might cause differences we see between populations. Thank you.